is Shikhar Mehta, and I am a bioengineering PhD candidate from Northeastern University in Boston. Osteoarthritis, or OA, is an extremely prevalent and debilitating disease of the joints that is primarily characterized by the degradation of articular cartilage, found at the ends of long bones in load-bearing joints of the body, resulting in significant pain and loss of joint functionality. Aging, the most common risk factor, can lead to the development of OA through the formation of advanced glycation end products, or AGEs, which are produced as a result of a Maillard reaction between sugars and protein lysine and arginine residues. In cartilage, AGEs form across long-lived proteins like collagen, forming crosslinks which greatly increase the stiffness and brittleness of the tissue, making it more susceptible to mechanical loading-induced degradation. Once formed, AGEs can also bind to their receptors RAGE on the surface of cartilage cells or chondrocytes. This triggers the upregulation of oxidative stress and inflammatory pathways, while reducing matrix biosynthesis, leading to the biochemical breakdown of cartilage. Currently, however, there are no in vitro models that accurately capture both the mechanical and biological response to AGEs and their inhibitors. Previous chondrocyte-based studies have failed to capture matrix cross-linking, while cartilage explant studies have utilized high sugar concentrations, which result in complete cell death. Thus, we sought out to create a live explant culture model that is representative of aged human cartilage that could be used to assess the anti-AGE properties of potential inhibitors. We first cultured bovine articular cartilage explants for 8 days with varying concentrations of ribose sugar to determine the optimal dose at which AGE crosslinks form while maintaining cell viability. Using this ribose concentration, we co-administered explants one of 3 natural anti-aging drugs, Resveratrol, Curcumin, and Eugenol. Following culture, we subjected explants to uniaxial, unconfined, stress relaxation testing for extraction of their mechanical properties. Explants were also assayed for AGE cross-linking, cell viability, metabolism, senescence, nitrite release, and glycosaminoglycan or GAG loss. We first determined that 100 millimolar ribose treatment induced a physiologically relevant 3 to 4 times increase in AGE cross-linking, which correlated to an increase in the equilibrium modulus of the tissue as well. This was suppressed by both resveratrol and curcumin, but not with eugenol. AGE cross-linking was also found to be inversely correlated to tissue permeability, where diffusion of GAGs was limited in response to decreased matrix pore size. We then used in silico blind docking simulations to determine the strength and locations of binding interactions between each of the 3 drugs and a type 2 collagen fragment. For each location, we multiplied the binding energy by the number of conformations and then divided by the total number of conformations possible along the entire length of a collagen 2 fragment to obtain a weighted binding energy, or WBE. We observed that resveratrol and curcumin possess stronger weighted binding energies than ribose and eugenol for the primary sites of glycation, where arginine and lysine residues were present, indicating the increased likelihood of these drugs to competitively inhibit AGE formation. We further determined that all 3 drugs were not cytotoxic at the concentrations used, and as an indicator of cell aging, chondrocytes were stained with beta-galactosidase. Consistent with previous results, resveratrol and curcumin suppressed ribose-induced senescence, whereas eugenol did not. In conclusion, we determined that AGE treatment of 100 millimolar ribose induced physiologically relevant mechanical and biological changes in cartilage through the formation of AGEs. We showed that resveratrol and curcumin, 2 naturally occurring anti-aging compounds, can competitively inhibit ribose activity and the formation of AGEs due to the presence of a hydroxyl group in the para position of their structure, which has an affinity for arginine and lysine residues. Thus, resveratrol and curcumin are effective therapeutics for inhibiting AGE formation and its downstream mechanical and biological activity, slowing OA progression for the aging population. We would like to make the following acknowledgements and thank our funding sources, the DoD and Northeastern Tier 1.